All right, so we'll launch into our first industry speaker session. I'd like to introduce Chantal Cadio first, a medical affairs scientist at Pfizer. So Chantal, if you wanted to take the podium, and I'll step just off to the side. Feel free to adjust, and there's the slide over.
That's good. Yes. So, hi, I'm a medical affairs scientist in rare disease at Pfizer Canada. So, very happy and grateful to be here to have this opportunity to present an update on our clinical program for the gene therapy in Duchenne muscular dystrophy at the request of Defeat Duchenne Canada. So, this is my disclaimer slide. So this presentation contains a discussion on a investigational product that does not have marketing authorization. So it's really for non-promotional purposes only. And it represents a scientific exchange. And because also of internal compliance, I cannot take any questions. So let's start. So Dr. McMillan already gave a good overview of what gene therapy is. But right, in essence, the objective of gene therapy is really to try to address the underlying cause of a disease. So in this case, right, the lack of a functional dystrophin. So if you concentrate at the bottom of the graphics at the bottom of the slide, so you identify, right, a copy of a working gene. So the DNA code, you take that gene, you put it into a package or vector or viral vector, and you take that vector containing the gene and you infuse it into the body. It's a one-time infusion, and then because of that vector, it targets the cells of interest. So in this case, the muscle cells, and then the muscle cells now contain this new blueprint. And so, the idea is that they would be able to then produce the protein that was missing or that had a mutation in it. So in this case here for this gene therapy program, it's a mini dystrophin protein that you would express. This is an overview of the DMD clinical trial program. It consists in 4 different clinical trials. So one are ongoing and one is planned. If I go like one by one, there's a phase 1B trial, and this recruited participants in the US only, 22 participants in total. And the objective was really at that point, it started in 2018, the recruitment is completed and all that, but the objective was really to try to identify what was the safe dosage to use, the appropriate dosage to use for the gene therapy. And this trial is still, we're still following the boys, we're still accumulating more safety information out of this. And now if we move to the phase 2 trial on this slide, so this one recruited participants in Australia and in the US. And this was for the early symptomatic boys aged between 2 to less than 4. And the objective of this trial was to address the safety and tolerability of the gene therapy. Then moving along, there's the phase 3 trial, which is the bigger trial. This one recruited participants in 13 countries in the world, so a global trial in ambulatory boys that were between 4 and less than 8 years of age. And this trial is really to look at the safety and efficacy of the gene therapy. And so there's no results yet for this trial, but we're waiting for those results. And then the last one, but not the least, is a planned trial in the non-ambulatory boys with no age restriction. So we're currently working with all the clinical experts to define more what that trial is going to look like, but it's one of the trials that are planned. So just 2 slides to go over the phase 3 trial in the ambulatory boys age 4 to 8, the one I just mentioned, which is the bigger trial. So this is an overview of the study design. So it's a global, so in many countries, in many centers, multi-center, it's randomized. It's double blind, so we don't know what the participants are getting. It's placebo controlled, so either the gene therapy or the placebo, they're getting one of the two, but we don't know which one. And the objective is to evaluate the safety and efficacy of the gene therapy in ambulatory male patients with DMD. And so there was a need, right, to have a placebo arm in this trial to be able to have clear results on the efficacy and safety endpoints. And also that's what the regulatory authorities are requesting. But there was a 2-to-1 randomization to allow to have less placebo upfront. So like 2/3 of the participants at the beginning received the investigational product and 1/3 received the placebo. And then they're followed for 1 year. And after 1 year, it was the reverse. So those that got placebo were able to receive the gene therapy. And then those that received the gene therapy received the placebo at that point. And then they're all being followed for 5 years. And for that same trial, this is an overview of the key inclusion-exclusion criteria on the left and the primary and secondary endpoints on the right. So if we start on the top left, the key inclusion criteria. So the participants were boys that were ambulatory between the age of 4 to less than 8 years of age and with a genetic diagnosis of Duchenne. And they needed to be on a stable dose of glucocorticoids for at least 3 months prior to the screening. And then just below that, the key exclusion criteria. They couldn't have some neutralizing antibodies against the AAV9 because if they had, prior to dosing, these neutralizing antibodies, this could pose a safety risk and could also affect the efficacy of the treatment. They were not able to be on the trial. They had received a treatment meant to increase the dystrophin expression within the last 6 months, or if they had received a gene therapy in the past. And there were also some mutations that were excluded, as Dr. McMillan mentioned, for safety purposes. And another exclusion was if they had a non-healed injury that may impact, the functional testing and assessments of the trial. So if we move to the right, the primary endpoint is the change from baseline in the North Star Ambulatory Assessment Score at year 1. And then there's many different secondary endpoints. Some of them are expression of the mini dystrophin and the distribution of it, change from baseline in CK levels, and then many different other functional outcomes. So this is just one slide to go over the phase 2 study I already mentioned. So this study is in the early symptomatic boys that are 2 to 4. And it's a single arm safety study, so there's no placebo in this study. The primary endpoint is really to look at safety, and then there are secondary endpoints to evaluate dystrophin, mini dystrophin expression, and some functional outcomes as well. So in summary, this is the clinical trial program that we have for Duchenne at Pfizer. So there's a phase 1b study to look at safety, then a phase 2 in the younger boys, also to look at safety, then the bigger phase 3 study in the ambulatory boys at 4 to 8 to look at efficacy and safety. We're waiting for those results. And then the planned phase 3 study in the non-ambulatory boys with no age restriction. So with this, I'm done. Huge thank you. Special, acknowledgement to the study participants and their families. for their time and dedication. And thank you.
Thank you very much, Chantal. I think we'll finish with all of the presentations, and then we'll take a question and answer at the end, if that's okay. So I'll ask Sarah Kahn, the patient experience manager for rare conditions at Roche, to come and speak to us next. And please, Sarah. Thank you very much. And that obviously advances the slides, and if you need to adjust the
do that. So hi, everyone. As mentioned, I'm Sarah Kahn, the Patient Experience Manager for Rare Conditions at Roche. It's been incredible hearing from the community over the past day or so. In particular, what resonated with me was Eric's message. He's really embraced his accessibility needs. He's having an improved quality of life with his friends and family, and our clinicians delivering their patient-centered care. So I'm quite pleased to be here and share with you who Roche is, what our work is, and the impact we strive to achieve for patients and their families. So at Roche Canada, patients and science are at the heart of everything we do. Having the courage to reinvent ourselves and question the status quo is what patients in the healthcare system expect from Roche, and our commitment today is as strong as it was on the first day of our Canadian journey in 1931. So today in Canada, we have almost 2,000 employees working across the country. Some are here today, and they're here to advocate and support the dialogue about increasing access to medicines and treatments for our rare disease community. So here you can see the impact we've had for patients treated with Roche medicines, tested with a Roche product, or in our trials. Partnering with the patient community to understand their everyday lives is essential to evolve the way we look at diseases. It is through collaboration with patients, their families, and patient organizations such as this one that we can personalize healthcare and develop better medicines and diagnostics. As a community, we know there's a great unmet need for Duchenne research. As a company, we're committed to advancing the research and science in the neuromuscular space for conditions like Duchenne. By studying the research and emerging science, we strive to improve our understanding of the nervous system and the different neuromuscular conditions. We engage and value our trusted neuromuscular clinical experts. We share scientific research, knowledge, updates in a timely manner with the appropriate experts so they distill it to the patient community as they see best. Our goal is to create a future of strength and independence for the neuromuscular community, and our mission is to make a positive impact on the lives of individuals within the Duchenne community. This is why we work closely with patient groups like Defeat Duchenne and Muscular Dystrophy Canada. So our work with the Duchenne community and patient groups has a strong role and voice in shaping our strategies and solutions for the community. This work isn't being done just in Canada, it's happening across the world through our different networks that help shape and inform the collective Duchenne voice. So to give you a few examples, my colleagues created a Duchenne advisory board made up of different patient groups, caregivers from a few countries across the world. And thankfully, a member of your own community, Nicola Worsfield, participated in this work and helped bring a Canadian lens to the different conversations we are having. We also collaborated on a Canadian patient journey with our global team. So this means we interviewed members from Muscular Dystrophy Canada and Defeat Duchenne Canada, along with clinicians in Canada, to better understand what patients are going through. We are all collectively partnering to understand what Duchenne families experience in their diagnosis, their interactions with the healthcare system, how their life is impacted, whether it's school, family, sibling. We want to know and understand what that is needed in terms of Duchenne education, supports, patient caregiver tools, and what resonates with the patient groups and their community. So this gives you a glimpse of the type of work that Roche patient experience team does and how important it is to hear directly from patients and caregivers at forums like this one to inform us and collaborate with us. So in 2019, Roche entered into a partnership with Sarepta, a biotechnology company based in the US, to collaborate on Duchenne research and gene therapy. Because we're passionate about advancing science no matter where it comes from, this means we seek out the most promising innovations in our labs or in the external environment in order to help patients and reduce the burden of illness and disease on society in a sustainable way. Only by partnering with the brightest minds in science and healthcare can we create breakthrough innovations and approaches that serve patients with difficult to treat conditions, both now and in the future. So at Roche, while we're focused on discovering and partnering on the latest innovations that will help patients around the world, we realize that in Canada, there's a significant challenge for any condition and treatment, and that is the amount of time it takes for patients to get access to new treatments. Roche Canada is encouraged by the recent federal investment in rare diseases with a hope that that investment will enable change in how rare diseases are evaluated, how decisions are made, how quickly access to treatments, testing, and supportive care is provided. Did you know medicines for rare diseases take an average of 2 years to reach people who need it the most? The dialogue many of you are engaged in and creating is vital for change. So we are in an important moment as it relates to healthcare in Canada. There's been an increased focus on driving this innovation for those diagnosed with a rare disease, and that focus is also being seen in the country. The announcement of the Federal Rare Disease Strategy in Canada highlights the unmet need, the need for advocates, governments, and stakeholders across the ecosystem to understand the value of these innovations, but also expedite the access to treatments and their diagnostics. Roche Canada is committed to working with patient organizations, key health advocates, patient thought leaders, or other industry partners, and government in increasing patient access to these new innovations. In addition, the great policy and advocacy work that Defeat Duchenne is doing, as well as the efforts of Canadian Organization for Rare Diseases, we hope to see the implementation of the federal government's commitment to rare diseases. We are truly encouraged by this, and we look forward to partnering with all levels of government, patient advocacy groups, and clinicians to ensure that the unmet needs of those impacted by rare conditions are addressed. Thank you for your time.
Thank you so much, Sarah. And I absolutely agree with you. I think with the federal government recently announcing the rare disease strategy, I think now is more timely than ever for there to be a push towards improving access, which has always been such an issue for us. Such an issue. And I think Defeat Duchenne is well positioned to push this ahead, either on their own or with other parent organizations for other conditions as well.
And I believe we just have a video from Catalyst before we introduce Dr. Hoffman.
Sure. Yeah. So yeah, please go ahead and play that. I'll maybe just sort of put it in context for 30 seconds, if that's okay. So we'll be meeting Dr. Eric Hoffman in just a moment. He'll be joining us online. Dr. Hoffman is the president and CEO of ReveraGen, which was the sponsor of the vamorolone trials, and Catalyst will be very important for the regulatory approval of vamorolone in Canada. So just to sort of put that in context, Catalyst will be moving things forward with regards to Health Canada approval of vamorolone in our country. So on that note, feel free to please roll the tape.
Hello and welcome. My name is Amy Grover, and I'm the head of advocacy at Catalyst Pharmaceuticals. Catalyst Pharmaceuticals is a innovative first-in-class medicines that address rare neurological and epileptic diseases, and we make it a priority to do so with exceptional patient focus. We have been involved in the neuromuscular area for over a decade as we developed our initial and flagship product, Firdapse, for Lambert-Eaton myasthenic syndrome. When that was approved in 2018 in the US, we proceeded to launch it in the US in 2019, and we subsequently obtained approval for it from Health Canada in 2020, where it was launched by Kai Pharmaceuticals, the company we decided to partner with in Canada. While we are new to this community, as we only obtained the North American rights to vamorolone in July 2023, we are working hard to make the connections within the Duchenne community to allow us to better understand and meet the needs of your community. While we have been primarily focused on doing so in the United States, due to its pending application for vamorolone at the time since approval as a Gamma in the United States by the US FDA on October 26, 2023, we are now in a better position and able to focus more resources on our engagement with Canada. So this is just the beginning. While I know many, if not all of you, have questions about those plans for Canada, we are not yet at a stage where we can provide much definitive information on key issues like timeline for approval or commercial availability. I can say that we are actively working on developing our plans in these important areas so that we can get to a point where we can discuss them and provide some answers to your questions. When we can do so, we would keep the line of communications open, and we will work with Defeat Duchenne to put the information out when we can. While I can commit today that Catalyst is just as interested as Synthera, our global on development activities to support Canada in continuing to supply the current clinical sites in Canada and the patients who are part of those sites, don't intend to change the current levels of supply or change the current clinical activities. They were created to support the product and patients and to develop data to be used to support the product's approval in Canada, and that is unchanged. While this is all I can say at this time, I do look forward to continuing this conversation over the long term as we continue to get to know you, your organization, and the Duchenne community, and you continue to learn more about Catalyst and how we support the rare disease communities in which we are active. Thank you so much, and have a wonderful rest of your program.
Thank you for running that. And I think Dr. Hoffman is joining us online.
Yeah, can you hear me?
We can hear you. Yeah, Eric?
Can you see me?
You yet, but are you joining with, there you are, there you are.
I can see me. Yeah, I thought this next session, we don't have any slides. You and I would sort of do this jointly as more of a chat. And you know, already it seems perhaps a bit disorienting because, as Hugh mentioned, I was the sponsor of a number of clinical trials in Canada, and Canada has been very key, actually central to vamorolone approval in the US, and it's also due to be approved in Europe on the 19th of December. So there's already been a positive opinion issued by what's called the CHMP a couple weeks ago. So it's expected to be adopted by EMA through Europe, and vamorolone approved throughout Europe in December. And what vamorolone was developed as sort of a collaboration between Hugh, many sites in Canada, the Cooperative International Neuromuscular Research Group, or Synergy, which Canada's been part of with Jean Ma since its onset, as well as other sites in Canada. And it's sort of run as a hybrid academic public-private partnership with a lot of government support from European Commission, from the US, and also FDA, NIH, Department of Defense, and also with foundations. So we worked with 15 foundations, including Defeat Duchenne. And in fact, there's an ongoing trial in Canada of younger children, 2 to 4-year-olds, and older children, 7 to 18-year-olds, of vamorolone at 4 sites in Canada. And that's supported by Defeat Duchenne Canada through a venture philanthropy agreement. So with this sort of hybrid approach, it wasn't really done by major pharma. ReveraGen is a very small company. It's 4 full-time people and 3 part-time people. And we really worked in this sort of academic / government / industry space. And with that, because ReveraGen is owned by nonprofits, and has arrangement with nonprofits, we never intended to do sales and marketing. So with that, as you've heard, we've partnered with Synthera through Europe, and then with Catalyst in North America to carry out the sales and marketing. So certainly Canada has been on the forefront of both vamorolone clinical trials and approval, and as mentioned, as you just heard by Catalyst, this will be continued access to vamorolone to the dozens, I think we're up to 70 or 80 kids in Canada treated with vamorolone long term. So just to give that little bit of background, and maybe to turn this into then a brief discussion with Hugh, and then answer any questions anybody has. Hugh, great presentation you gave earlier, and great comparing drug programs in the US versus Canada. I would just maybe begin the discussion because I thought 1 or 2 aspects that maybe I would emphasize a bit more is you mentioned the accelerated approval process in the US in passing, and how there's a number of drugs approved in the US that aren't approved in Canada. But to point out that all those drugs that are approved in the US and aren't in Canada are not approved in Europe as well. So Europe does not have any of the exon skipping or the gene therapy drugs approved like Canada. And so really Europe and Canada seem more closely aligned. And that has to do, I think, with you. Again, you mentioned this sort of health technology assessments program. I called it, I forget the name of the organization in Canada it's called. But that health technology assessments process is shared by Europe and Canada and is not shared by the US. And that has a fundamental effect on the drugs approved, because under this accelerated process, which is unique to the US, that's a biomarker approval. You don't have to show clinical benefit in order to get a drug approval under the accelerated process in the US. That's done post-marketing through additional trials. Whereas Canada and Europe and most of the world does not have a similar biomarker-based process. Instead, under HTA, health technology assessments, you really need to show clinical benefit and have evidence of clinical benefit or efficacy. And because none of those 5 drugs in the US have yet shown or proven clinical benefit, it's really why I think a primary reason they're not approved in Europe, and similarly an impediment to getting them approved in Canada. And you might ask, your audience might ask, well, this health technology assessment, what is it and why doesn't the US have it? It's because the fundamental nature of the healthcare systems, where in the US, it's a market-based system where drug pricing and access is what the market will bear. So there is not an HTA process that looks at value to the healthcare system. Whereas the HTA process, because it supports nationalized healthcare systems in Canada and Europe, that process needs to show that the new drug will bring value to the healthcare system and to the patients in that nationalized healthcare system. So I would argue that that might be a major difference between the access that you mentioned in passing, but I'd like to hear your reaction to that. If you think I'm being overzealous in blaming HTA in a way.
No, I agree with what you're saying, Eric, and I think that it is industry partner can file for regulatory approval at a much earlier stage in the US because of them allowing a surrogate marker to be used. Whereas, as you mentioned, in Canada, we need to have more evidence of clinical efficacy for that approval to take place, both in Canada and also in Europe as well. And I thank you for pointing out that common feature between Canada and Europe. And then as part of the review process with CADITH, just the value for money piece and the expenditure associated with a disease or a condition versus how that may change when a product is added. That certainly is something that CADITH will take into account when they're making decisions around reimbursement for provinces. And obviously it takes time for that to happen, and a lot of data needs to be presented to crunch the numbers about how the use of a drug might change healthcare expenditures and value for money, which is not part of the discussion, at least to my knowledge, in the United States at the FDA level.
Right. And that tees up a next point regarding anticipated approvals, and specifically regarding vamorolone. Vamorolone, in large part because the participation of yourself and many Canadians in the vamorolone trials, we carried those out double-blind, placebo-controlled in 32 sites in 11 countries, and that has shown clinical benefit. So vamorolone is not approved under the accelerated pathway. It's approved under the traditional pathway by the FDA. And it will become in December, presumably, because it's, as I mentioned, it's not quite approved, but it's already slated through this positive opinion. Will be the first drug receiving full approval for Duchenne in Europe. Why that's important is because it has shown clinical benefit. So then that tees up the CADITH approach much more cleanly in a way. So in other words, it's a straight path through, as you mentioned, the Canadian HTA process. And so it should hopefully be relatively smooth sailing, as so far it's been in Europe through that process. So which brings up another thing that maybe could be mentioned, which is a little trickier, but is the fact that because the US is under this market-based what the market will bear system without an HTA process, the drug prices are higher. And that's, you've heard, I'm sure, all throughout your papers, including, talking about bringing buses of US patients up across the border into Canada. Because the market in the US system will bear higher costs, in part because probably there's not the same HTA process that keeps needs to justify the costs. Well, that becomes, when in a new drug, that can become also challenging in approvals, because the US approval, which is focused on what the market will bear, will get out ahead for purely commercial reasons than the Canadian. So, just because you share that border, that becomes another tricky thing. So I think, in summary, it will be smooth sailing, hopefully, with the HTA process and vamorolone, and everybody's looking at that. But it's just there's other factors that are in that mix. Perhaps you could respond to that?
Yeah, no, I agree, Eric. I agree with everything you said. I absolutely do. We'll also have to bring it to a close just after this point, because there'll be time at the end, and I'm very hopeful you'll be able to stay online, Eric, and we'll be able to unmute you to answer any questions that come up with vamorolone. But I'm very grateful for everything you shared, and specifically emphasizing some of the differences between the US and Canada in the approvals process, because I as I'm sure you'll hear later, that has been a challenge and a frustration for many families that are noticing that discrepancy in the context of vamorolone, like we have with other drugs as well. So thank you so much, and please stay tuned. Don't leave us. And very last is I'll invite Aaron Novak, who's the Executive Director in the Global Medical Affairs team at Dyne Therapeutics, to speak to us. The floor is yours, Aaron.
Macmillan. So yeah, my name is Aaron Novak. I work in the medical affairs group at Dyne Therapeutics, and I'd just like to thank Defeat Duchenne Canada and everyone for having us here today to give you an update on our Dyne 251 program, which is our clinical candidate for people with Duchenne who have mutations amenable to 51 skipping. Oh, sure, sorry. So here's some of our forward-looking statements. I'll be making some forward-looking statements, and I'll also be talking about an investigational therapeutic. If you want to see these more, if you need a sleep aid or anything, they're on our website. Okay, so I start this slide, presentation with our mission. At Dyne is really the focus of everything we do, which is to provide life-transforming therapeutics for people living with serious muscle diseases. And today, as I mentioned, I'll be giving an update on our Dyne 251 program. I'm really excited to inform you that we've started dosing that program. We have some exciting updates that I'll be sharing in a little bit. So to start, I just talk a little bit about our FORCE platform. So our FORCE platform really drives everything that we do as far as it comes to developing and delivering muscle therapeutics for serious muscle diseases. It's really built on this idea that we need to overcome one of the major unmet needs and challenges of delivering therapeutics in muscle diseases, which is delivering it to the muscle, not only just any muscle, but muscles that are implicated in the disease, for example, skeletal, cardiac, smooth muscle. So that's really how it was developed, really trying to overcome those challenges of getting it in a sufficient amount into those important muscles. And so our FORCE platform is really built up of three components. So I'll go through each of the components, and then I'll come back at the end a little bit and just talk a little bit about why we think it's important that we're targeting a certain receptor called the transferrin receptor type 1. So you can see the three components are our antigen binding fragment, which targets this receptor that's on cells, on the surface of cells, called transferrin receptor type 1. We have a linker, which links that to the antisense oligonucleotide of choice, depending on the disease that we're working on. So really, those are the three components. So I'll start first with the Fab. So you can see there, kind of in the bottom left, there's that little gray structure, looks like a Y, which is the monoclonal antibody. We've chosen just a portion of that monoclonal antibody, which is the antibody fragment. We did that because we think it provides potential benefits over including the entire monoclonal antibody, which includes smaller size, lower protein load altogether, and potential for improved safety profile. And really, the goal of the Fab is to deliver, as I mentioned, these antisense oligonucleotides to the important muscles. Next, we have the linker. So the linker is what links. It's a valine citrulline linker. It's a linker that's been used in a number of other antibody drug conjugates that have been approved. So we think that again kind of helps to de-risk some of the issues with with our FORCE platform. And then lastly, we have on the bottom, looks like the comb, little old school comb there that Dr. McMillan mentioned. In this case, the antisense oligonucleotide. And so the nice thing about the FORCE platform is really the versatility and the modularity that it allows us. So the Fab and the linker are the same across all of our programs. We've got a number of other programs for other muscle diseases. And what we're able to change is this ASO at the bottom. And so really we're able to target the biology that we're looking for in each of those different diseases. We're able to tailor it to target those therapies. So for example, in Duchenne, where we're looking to increase exon skipping and therefore dystrophin, we've chosen an antisense oligonucleotide, a phosphorodiamidate morpholino antisense oligonucleotide. It's a mouthful, so it's called a PMO. So we've chosen the PMO to really get into the nucleus and deliver dystrophin to those key muscles. And so now let's talk back a little bit about transferrin receptor 1. So transferrin receptor 1 is, and it's really where it sort of differentiates our platform from some of the other next generation antisense oligonucleotides, is that we're really harnessing the cell's natural biology. So TFR1 is already expressed across muscle cells. And so really what we're doing is we're using it to deliver through this antibody-mediated delivery of the Fab, which brings in our exon skipping. So it's really like a key. When it binds to the transferrin receptor, it sort of allows the exon skipping therapy to enter into those muscles and be able to deliver to those key muscles. All right, so on to the next slide. So This slide and the next slide, just a little bit of information at the sort of as we went into the clinic, we had to do a lot of work to test our molecule in different animal models to make sure that we were going to not only have a safe therapeutic, but something that's going to provide a benefit, hopefully. So we haven't really shied away from exploring all the different tools and all the different the models that we have within Duchenne research. And so you can see some of them listed here. So I'll start from the bottom. We've used non-human primates or monkeys to look at both the safety and the exon skipping of our therapeutics. In this case, we can actually use Dyne 251. Non-human primates are not a disease model, so we can't actually look at dystrophin, but we can look at the safety and we can look at how much exon skipping is produced in that model. Next, we've used a common model, the MDX mouse model, which is used across much research within Duchenne. This allows us to look at exon skipping, dystrophin, as well as function in the mouse. We've also looked at in vitro models of human cell lines. So I'll just show you one slide here in the mouse model. So just to orient you, this is the MDX mouse model. So MDX mice have a mutation in exon 23. So in this case, we're using a tool molecule. So it's force linked to a exon skipping that binds and skips exon 23. And so what we did is we gave the mouse a single dose of 30 milligrams per kilogram. And after 4 weeks, we look at this data. So this is 4 weeks after a single infusion with this force conjugate. And you can see across the top, across those key muscles of quadriceps, diaphragm, and heart, we see significant dystrophin expression. So about 51% in the quadriceps, about 90 in the diaphragm, and about 77 in the heart. And I think yesterday's presentation was really great because it really, I think, drove home the importance of treating both the lungs as well as the heart. And that's one of the challenges with current or sort of the older first generation exon skipping is they don't really get there to those muscles. So we're really excited that we see exon skipping and dystrophin in those key muscles. On the bottom, it's what's called percent positive dystrophin fibers. Dr. McMillan went over that a little bit. It shows that the dystrophin that you're producing is getting to the cell membrane and actually doing what it's supposed to do. So it's sort of an early sign that what you're producing might have some functional benefit. And you can see there, again, we see 68% positive dystrophin fibers in quadriceps and about 80% across the diaphragm and the heart. All right, so on to our DELIVER study. So the DELIVER study is a phase 1/2 trial. And again, I think it's really helpful. Dr. McMillan kind of laid out what a phase 1/2 trial is. So again, we're looking at early first-in-human safety as well as some efficacy endpoints. So our phase 1/2 trial is a global trial. It's a double-blinded placebo-controlled trial. So it means that some patients received placebo, and double-blind means that neither the patients, the hospitals, or ourselves know what the patients are receiving. And so we are enrolling approximately 48 boys between the age of 4 and 16 years old. We're allowing both ambulant and non-ambulant patients into the trial. So the trial really is 4 parts. So there's a 6-week eligibility period where they do some screening. They do some of the screening endpoints to see if they meet those inclusion-exclusion criteria. Then there's a 24-week multiple ascending dose period. And this is a period where participants receive either Dyne 251 or placebo, and they're receiving it every 4 weeks. So again, that's another one of these benefits of the FORCE platform is we can hopefully dose less often. So for 4 weeks, they're receiving the therapeutic. There's this multiple ascending dose means we start at a low dose, we do that. If it's safe, both an internal and an external consultants decide that they can go to the next level, and so on and so forth. So they move up levels as the therapeutic is just determined to be safe. So after that's 24 weeks, they get into an open label extension period. In that period now, anybody who was on placebo in the MAD portion now gets to transfer over and receive active therapeutic. So by the time after 24 weeks and on, everyone is receiving active therapeutic. So there's a 24-week open label, and then there's a 96-week long-term extension study. As you can imagine, patients are in different phases and different cohorts. And so patients who started a low cohort, once it's determined to be safe, can be brought up to that next sort of highest safe dose. We have some key primary endpoints. You can see their safety and tolerability, as well as change from baseline in dystrophin production. We also have some key secondary endpoints that are looking at pharmacokinetics. So that's the way the drug gets into the body, moves around the body, and then is excreted from the body. We also have some important functional assessments, looking at the North Star Ambulatory Assessment, as well as the PUL 2.0. And that gives us sort of a assessment of both upper limb function and lower limb function. We also have the stride velocity 95 percentile, sort of a newer assessment where participants wear ankle or arm bands, sort of like an activity monitor for a number of days, and we're able to see that sort of continuous activity. And we're also monitoring, important cardiac and respiratory function as well. So the newest announcement that we just made last week was that we've now dosed 32 patients in the trial. We've gone up through 5 cohorts. So we just finished enrolling the 10 milligram per kilogram cohort. And now we're currently enrolling the 20 milligram per kilogram cohort. So it's been nice to see that we've been able to, with the safety profile, be able to go up through those multiple ascending doses and now starting to give a much higher dose. And we hope to have initial safety and tolerability as well as dystrophin data in early January 24. So hopefully next year, we'll happy to come back and talk about the data once it's out. Okay, so just some key inclusion-exclusion criteria. This can be many more of these, as well as much more detail on these inclusion criteria, as well as the other endpoints I didn't highlight, can be found on clinicaltrials.gov. It's super small on that slide, which you won't be able to read, but in the back, there's these little handouts that has the ct.gov listing on there. So, or you can just search Dyne 251 clinicaltrials.gov, and you can find it there. So as I mentioned, 48 boys between the ages of 4 to 16 years old. They have to have a mutation that is amenable to 51 skipping. There is a biopsy in this trial. So we do a biopsy at baseline and 24 weeks. As I mentioned, ambulatory and non-ambulatory boys are able to join. They have to be receiving a stable corticosteroid dose, a steroid dose for 3 months prior to the start of the trial, and maintain that throughout the trial. There's certain cardiac and respiratory assessments that they have to meet in order to be included. They cannot have received gene therapy, because gene therapy is a one-time therapy. You can't undo it. So we don't want to. We're trying to test the impact of our therapeutic without any confounding effects. However, if patients were on exon skipping therapies prior to this, they can wash out and get onto our therapy. So it is a global study. It's being done in the US, in Europe, as well as in Canada. So we have 2 sites in Canada right now. We've got one here, Dr. McMillan in Ottawa, and Dr. Campbell in London. We hope to have another site in early January. And just to end, we're really thankful for being here. We take up every opportunity we can to talk to the community to understand what your needs are, as well as how we can help. So thank you again for your voice, your opinions, your advice. And if you have any questions, happy to answer. Thank you.
Thank you, Chantal, Sarah, Dr. Hoffman, and Aaron for your presentations. Much appreciated. Any questions online or in the room?
I think what we'll do in the interest of time is the questions that came through on the Whova app, we'll have the speakers answer on Whova directly. But if there are any questions in the room, we have time for a couple.
Thank you for the great presentations. So I just wanted to follow up on the question that was kind of asked earlier, but I wanted to ask now to you guys. So for Dyne Therapeutics, for instance, for the trial you have ongoing, you mentioned that the stable corticosteroid dose was required for 3 months prior and throughout. Are you thinking about vamorolone in future? Is that going to be a possibility for a different inclusion criterion?
Does this work?
Yeah, I think it does, yeah.
Yeah. So yes, as I think as Dr. McMillan mentioned, we've been knowing, we knew this was coming, so we've been thinking about it. Obviously it was just approved, so we were waiting to see the label and to sort of see how that will work out into our clinical trials. So we're still taking that in. As Dr. McMillan mentioned, it's likely that it's likely that we will allow it for places where it's approved, but where it's still an investigational therapy, we have to probably wait for it to be approved in that region.
Thank you all for the presentations. I was just wondering about the AAV antibodies. I know it's an exclusion criteria. Can you comment on the prevalence of it, or if there's any factor that may lead to someone being exposed, or is there, any way, maybe not to predict, but yeah, just have a sense of it.
Yeah, no, absolutely. I don't know if Sarah or Chantal want to answer that, or I could take it if you don't. I'm fine. So I think we're learning more and more about that, Alfred. With SMA, the AAV9 seroprevalence was in the ballpark of about 5% of patients. Some of that was transmission from mom to babies that would then clear over time. The older we get, the greater the likelihood that a person may be seropositive. So we're learning more about it as it's tested in older patients. The one difference with the adeno-associated virus is, even separate from the clinical trials, and just thinking about AAV virus as it exists in nature, it can't actually cause disease in itself. It only causes disease if there's a co-infection with an adenovirus as well. So that's one of the things that make it also less prevalent in the population, because someone would have a particularly, quote, bad cold where they have an adenovirus and an AAV infection superimposed on that. So the majority of people are antibody negative, but we're learning more with time about what the seropositivity rates are in different populations with different ages.
Thank you.
Pleasure.
Any other questions in the room? Okay, if not, I think we can wrap up the session.
So just on behalf of Brian and Defeat Duchenne Canada, thank you to our industry partners for joining us in person. It's really wonderful to have you here with us.